Today's lecture is on DNA. And in addition, what's really important is not just that I sort of convey facts to you and then you learn sort of what makes DNA and that we know it's the genetic material and things like that, but how we came to know these things. I mean, part of what you should get out of a course like this is not what scientific knowledge is, but how you get it, how you do experiments and figure things out. So through today's lecture, there are going to be some good examples of experiments that are now classic, very important experiments in the history of biology that I'm going to show you not just the results, but what the experiments were, so you get a sense of that. Okay. As I said last time, I start each lecture with a set of questions just to orient you of what we're going to cover each time. And so the questions we're going to cover today are, what is DNA? How do we know that DNA is the genetic material, which I'm assuming all of you sort of came to this class already in some way knowing. And then the final question is, how is DNA copied? And that's related to this question because obviously for something to serve as a mechanism of inheritance, to be a genetic material where progeny inherit something from their parents, that material has to be copied somehow. Okay, and so what we're going to finish with today is just starting to talk about how DNA is copied and therefore serves as a genetic material. So, as I said, probably before any of you entered this class, you could recognize that this was DNA. It's sort of an iconic shape and visual now in our culture. It's this classic intertwined helix, double helix shape, okay, with a ladder of steps joining the two helices, okay? And the reason you probably know that that's true is either you were paying attention in high school when you took biology, or because you're just sort of a person in our culture in the year 2010. And so I've given you some examples here of the kind of ridiculous heights that it's gone to in terms of how much just this sort of iconographic image of DNA has entered our culture, okay? So, for example, you can buy DNA perfume, right, with the fancy, you know, DNA pour femme, right? You can buy DNA earrings. Even when you buy them, you're told what part of that earring corresponds to which part of the DNA, okay? And we'll get to in the lecture, you know, what adenine and phosphate and all those things are. People spend their time making DNA out of Legos, okay? And somehow that has to do with building a stronger tomorrow. This is one of my favorite examples. So this is a logo for a company called Perpetua, okay? And so it's a DNA, obviously double helix, inside of a heart, okay? Anyone have a guess of what this company does or sells? Drugs, that would be a good guess. That's actually not true. No, much more mundane. Okay, here, I'm going to give you a little bit more of their literature so you can guess better, okay? That's a necklace, okay? You can see the spiral, of DNA inside the little capsule, okay, right here. You can see that it's sort of helices, nice colored ones. You can pick your favorite color, right? So you can make jewelry with your loved one's DNA in it. Okay, so that's sort of what the heart represents, right? That it's, you know, someone special to you and you're holding their DNA. Okay, but that someone, right, is not a human being, it's your pet. Okay, and that's sort of, you know, like the clever name, right? Perpetua, right? So what you do is you take like a big Q-tip and you go inside your dog's mouth and go back and forth. And you're actually going to do this in next week's lab with your own mouth, you know, scraping cells off of your cheek, right? And that's a good way to get DNA. And you send it to the company, and they isolate the DNA out of those cells, and they turn it into this fancy little structure here. They encapsulate it, and then you could wear it. And that sort of keeps your pet, you know, close to you at all times. And I guess if your pet runs away, you have some way of maybe identifying it when you try and get it back. So people are kind of into this, I guess, because the company still exists. I found this, you know, 3 years ago, and the company still exists, so they must be doing good business. it turns out that the start of DNA research was not so glamorous as what it's become today in being perfume and necklaces and so on. It started with a guy named Friedrich Miescher, who was a Swiss biologist. And he was interested in the structure of the nucleus. So ever since microscopes were powerful enough to look at cells, people realized that cells were not just sort of uniform structures, but if you look closely at a cell, you could see that in the center of it, center of your cells, for example, there was a sort of discrete body that was called the nucleus. And he was interested in what that nucleus actually was. And so he wanted to isolate a lot of nuclei and figure out what the chemical structure of those nuclei was. And so he figured out a clever way to do that. He realized that white blood cells contain, are sort of a readily available source of DNA. He didn't know it was DNA at the time. It was a source of nuclei. And the best place to get them was the hospital down the street. Okay, and because they would just throw out all their bandages when the patients were wrapped up, and then the bandages were thrown away, and the bandages had all this pus in it, and that was mostly white blood cells. Okay, so what he would do is he would collect all of these bandages, get the cells, isolate the nuclei from the cells, and then do chemical experiments to try and figure out what those nuclei contained. And this was a long time ago, right? This is right after the Civil War. Not quite modern science. And so the best he could do at that point was to find out that the structure in the nuclei contained a lot of two elements, phosphorus and nitrogen. Okay? He called the substance nuclein, and we now know that what he was studying is DNA, which is deoxyribonucleic acid, and we'll go through why it's called this long name right now. So in, I think, 2 weeks in lab, you're going to isolate DNA in a manner very similar to what Miescher did. Okay? You're not going to go down to the hospital and get bandages. We're going to use some other source of cells instead. But it turns out that it's actually fairly easy to isolate DNA. All you need really is a margarita. Okay? So the three things you need to isolate DNA are acid, salt, and alcohol. Okay? So as you'll see in lab, that it works pretty easily, and anyone could really isolate DNA. Okay, so what is it? What is the chemical structure of DNA that we now know, building upon what Miescher first found out? DNA basically has three parts to it, okay? And so I don't want anyone to get scared when I put a diagram like this up. I will never, ever, ever ask you to say, draw this, on a test, say what's the structure of DNA, and expect you to know all these chemical bonds and everything. But you should be able to sort of look at this and interpret it a little bit with my guidance, obviously. So you've probably all seen diagrams like this before from, say, high school chemistry or something, right? It's just a diagram showing atoms in the molecule that are connected in the ways they're connected. And so you have carbons and hydrogens and oxygens and phosphorus and nitrogen. Okay, so there's the two things that Miescher found that were sort of unusual about DNA. And we can break this down into just three basic parts. So the first part is a sugar. Okay, so this is a classic 5-carbon sugar, 1, 2, 3, 4, 5, and it's called deoxyribose. Okay, so that's part of where the name comes from. It looks a lot like any old sugar, so like a sugar that you would be familiar with, like glucose, okay, would be similar, except instead of a pentagon, it would be a hexagon because it's a 6-carbon sugar, okay? So a big part of DNA is this sugar, right? The second part of the building block of DNA is a phosphate group, okay? So that's a phosphorus with 4 oxygen atoms. And as you'll notice, the phosphate has negative charge, okay? That's important. That's what makes DNA an acid, okay? Deoxyribonucleic acid. And it also has important properties, as you'll see later in the lecture, in terms of how DNA is stored within a cell. And then finally, it has what's called a nitrogenous base. Nitrogenous just because there are a bunch of nitrogen atoms. It's this part, actually, that's the most important part of DNA, because this is where the language of DNA is written. Okay, there are 4 different flavors of nitrogenous base, and depending on which one you have at a particular place in your DNA molecule, that's sort of the alphabet of DNA. Okay, and I'll show you that in a second. Now, so this is one repeating unit of DNA. So what happens when you make a long DNA molecule, so like your chromosomes, which have millions of DNA of these units in a row, is that the phosphate of one of these gets connected to the sugar of the next over and over and over and over again. Okay? And so sometimes that's called the backbone, the sugar phosphate backbone. And then these bases are sort of sticking out the side, and that's the alphabet. So the backbone really isn't that important to us in terms of the information content of DNA. The only thing that's important to us in terms of the information in DNA are these bases. And so as I said, the bases come in 4 flavors. They're called adenine, guanine, thymine, and cytosine, okay, abbreviated as A, G, T, and C. Again, I don't care that you know which one is which or what the exact structure of these are. The really important thing is that there are 4 types. There's 4 letters, basically, in the DNA language, and these are those letters, okay? And in fact, if I went upstairs into one of the biology labs and I asked someone to draw me adenine, they probably couldn't, okay? And the reason is, as practicing biologists, many of us are not really interested in this level of detail. What we're interested in is the information content, and for that, it's sufficient to know that there's A's, G's, T's, and C's. Okay, so this is what DNA looks like. Then the question becomes, well, how do we know that it's important? How do we know that DNA is the thing that carries genetic information in it? And there's a long series of experiments that kept building up the story until there was final proof of that, and that's what I want to go through now. Both because it's how you start learning about this stuff, but also because there's an interesting history of science to it. Of how things that are obvious to us now once were not obvious. And part of being in a science class is trying to transfer your brain back to this time when things weren't obvious and how people were able to figure them out. Okay, step 1, how do we know DNA is the genetic material? The first piece of evidence is that chromosomes are made of DNA and protein. Okay, so what does that mean? Well, as I said, ever since microscopes became powerful enough to look inside cells, people realized that there were interesting structures called nuclei, and that sometimes those nuclei became even more interesting, in particular when the cells were dividing, because instead of just having a round blob in the middle of a cell, you saw these sort of finger-like structures that came to be known as chromosomes. Okay? And it was obvious that these chromosomes took shape when the cells were in the process of dividing, so making 2 cells where there used to be 1 cell. Okay? And so just by that observation alone, it seems that chromosomes might be important for contributing the information and keeping the information going. Okay? Chromosome, by the way, is constructed from the roots chromo and soma, colored body. So these nuclei and chromosomes were just the things that stained a dark color when they were viewed under the microscope. Okay, so then it was discovered that the chromosomes, which it seemed like had something to do with inheritance, were made of DNA and protein. Okay, so those were the 2 major guesses at the beginning of this century, last century, let's say, for what was the genetic material. It was either DNA or protein, and actually, as you'll see later in the lecture, most people bet on protein. Okay, most people were wrong about what the genetic material was. Okay, next piece of evidence. Chromosomes carry genes. Okay, so as I said, just looking at chromosomes under a microscope, it seems to suggest that maybe they have something to do with keeping genetic information going as cells divide, but it's not proof that they do. Okay, and it wasn't until the early 1900s that it was shown that the chromosomes really do have something to do with inheritance. Okay, we're not really going to talk about genes today. I'm not going to define what a gene is today, but I think you all probably have a basic enough understanding of what a gene is to get through today's lecture. Okay, so how many of you have heard of Gregor Mendel? What did he study? Pea plants. What did he find? So he found that some plants look different from others, right? And that if you cross two plants that were different to each other, then sometimes you got a plant that looked like one parent, and sometimes you got a plant that looked like the other parent. And if you cross those progeny among themselves again, you saw, again, the different types in their progeny. And from that, and we'll talk about this later on, and so this is meant to be just a very quick review. From experiments of just crossing pea plants, he was able to deduce very, very, very crucial information about genes. Namely, that each parent contributes one copy of each gene to the progeny, right? And that some of those versions of the genes are dominant and some are recessive. So if you have two copies of each gene, it's the dominant one that gets turned into how you look as a pea plant. And the basic rules he discovered about genetics basically apply to every organism we know. Okay, and so this is a picture from your textbook of the eye color of different flies. Okay, and so there's a gene that's required for fly eyes to be red. You could have a mutant version of the gene that makes the fly's eyes white. And by crosses, you can tell that those genes sort of maintain their integrity even in the progeny of, say, a white-eyed fly and a red-eyed fly that's red-eyed. That white-eyed version of the gene still exists and could be passed on to progeny again. So that's all genetics, right? And what was really required then was some correspondence between genetics, doing crosses and following whether the fly's eyes were red or white, and chromosomes, okay? Because there were these physical structures you could see under a microscope, and then there were these abstract notions of genes that were controlling things like eye color. So in the early 1900s, these two things were put together, okay? And they were actually put together up the street at Columbia, okay? There was a famous laboratory there led by a guy named Morgan, okay? It came to be known as the Fly Lab. And they did a lot of experiments crossing flies together, and they figured out a way to look at fly chromosomes. And by finding really rare exceptions to the inheritance rule, so when Mendel did his crosses, there was a regularity to it, and he could then predict what kind of pea plant he would get from a cross. The same thing in flies. If I cross a red-eyed male and a white-eyed female, I sort of know what to expect in the progeny. From that, you can get rare exceptions. And what the group up at Columbia was able to do was to find those exceptions. They studied a large number of flies, find those exceptions, and show that in the cases where there was exceptions, the chromosomes were going the wrong way. Okay, so when the cells were dividing, instead of getting one copy of each chromosome in each of the daughter cells, two copies went the same direction. Okay, and the chromosomes were not sorted out properly. Okay, so this is what their experimental results look like. This is not an experiment we're going to go through in detail. There are other experiments today we're going to go through in detail. You can see just from figure whatever of their paper that it was a very, very complicated series of experiments. And the only reason I'm showing you is to point out how complicated it was. Okay? And the other reason I'm showing it to you is that their proof that genes are carried on chromosomes was the first paper in the first issue of a journal called Genetics. Okay, and it was published in 1916. So if genetics were a religion, and this journal was the Bible, right, this would be Genesis chapter 1, verse 1. Genes are carried on chromosomes. Okay? So far we have chromosomes contain DNA and protein. Chromosomes carry genes. Where do we go next? We go to 1928. And in the 20s, after World War I, there were a bunch of pneumonia epidemics. And a guy named Frederick Griffith, who was an officer in the British Army and it was a doctor, was trying to develop a vaccine against a major cause of pneumonia, which is a Streptococcus pneumoniae bacteria. And he discovered something very interesting in his experiments. And we're going to go through his experiments because they're a great example of what we like to call in science an elegant experiment. Simple, clear-cut, and giving a really informative answer. Okay? And so what he discovered is that, there's a chemical substance that can be inherited, okay? And the way he did this was as follows. He found that there were two types of Streptococcus bacteria, okay? He called them smooth and rough, and what was different about them was how they looked when they were cultured on a petri dish, okay? So the smooth colonies had this sort of glossy, smooth and appearance, and the rough ones are smaller, and if you could magnify this up, you would see that they had sort of rough edges instead of the smooth, glossy appearance. And so some of you might not have seen cultures of cells growing on a petri dish, so I brought a petri dish. Okay, this is not Streptococcus, don't panic. This is E. coli, so each one of you already has about 1 trillion of these sitting inside your intestines, so this is not going to harm you. But I just want to give you a sense of sort of what he was looking at and what we're looking at when we say there's a colony of a particular bacterium on a plate. Okay, so here's what you do. You have this jello, basically. Imagine like chicken soup jello. Okay, that's basically what the culture medium is for bacteria. So you can see that it's not liquid, it's solid, but it sort of has this like yellowish color. And what you do is you put down cells on this plate. And each colony here is millions of cells that are descended from one cell each. Okay? So you put down cells that pretty sparsely, and then each one divides and divides and divides. And at first you can't see them, but if you let them grow until there are millions of them, then they actually have this macroscopic appearance on a plate. Okay? So I'm just going to sort of quickly go through. I'm not going to pass this around just in case. Okay? But I want you all to see what sort of colonies on a plate look like. And in lab, in a few weeks, you'll actually get to culture your own colonies from the environment, by which I mean the doorknobs and bathroom handles from around here, and see what grows. Okay, so can you guys see too? Right, so basically little dots. Each one of those contains millions of cells, right? Millions. So one cell is tiny. It's about 1/1,000,000th of a meter in diameter. Okay, can you guys see? Right. So this plate has, maybe 1,000 colonies on it. Each colony has, millions of cells in it. Right. So sometimes you go to a doctor and you get a sore throat. They take some kind of Q-tip thing, stick it in your throat, rub it on a plate like this, and see what grows. Right. And if something grows that looks like that, you're in trouble. Right? Everyone get a chance to see it? Okay, so this is the kind of thing he was doing. He was doing something a little bit more sinister, too, because he was then taking these cells and injecting them into mice. Okay, and you'll see how he went about doing that. So here's the experiment. He had these two different forms, the smooth form and the rough form. The smooth form is smooth because it has sort of a coat on it. It has made up of polysaccharides, sort of like starch. And the rough one lacks that coat, so it's not as sort of shiny. And it turns out that the smooth one is the dangerous one, and the rough one is not dangerous. And the way you know that is you have your rough cells, you pick them up and inject them into a mouse, and the mouse is still alive. If you do the same thing with the smooth cells, you pick them up and inject them into a mouse, the mice dies. Okay? Very quickly. They basically get pneumonia and die. Okay? So here's what Griffith did. He took these smooth cells, and he transferred them to a test tube. Okay? That's my depiction of a test tube. And then he heated that test tube to kill all the cells. How does he know he killed all the cells? He takes those cells and then injects them into a mouse. If all the cells are dead, then the mouse lives. Right? Everyone with me so far? Okay? So that's what he's done. He's killed those smooth cells. He does the same thing, takes the smooth cells, heats them up. Instead of injecting them straight into a mouse, he mixes them with some rough cells. Okay? So think about this. Smooth cells, heated up, are dead. They can't infect a mouse. Rough cells, not heated up, can't make a mouse sick either. Okay? But what happens is he takes this dead material from the smooth cells, mixes it with the live rough cells, injects it into a mouse, and the mouse dies. Okay? So what does that mean? It means that there's something in this test tube that's a physical substance, a chemical substance, not a living thing anymore, that can transform rough cells into smooth cells. Okay? And he called it the transforming principle. Okay? So there's some material in this test tube after the cells have died that transforms rough cells into smooth cells. Okay? And so he can test this, right? So all this shows really is that rough cells plus dead smooth cells kills a mouse. Okay? So how does he show that he's actually transformed them into smooth cells? Well, he takes this mouse, he gets its blood, he gets the cells out of the blood and puts them on a plate again. Okay? So you take the dead mouse, you put the cells back on a plate, and what does he see? Smooth cells. Okay? And that's because they really have been transformed. He's taken rough cells, mixed them with something from the dead smooth cells, and made smooth cells again. Okay, so at this point, we still don't know what this substance is. All we know is it's a substance. Okay? And what that allows then is later scientists to do chemical experiments to figure out what that substance is. Okay? And that happened a few years later, also up the street. Okay? This time east side instead of west side at Rockefeller Institute, which is now called Rockefeller University. Three scientists, Avery, McLeod, and McCarty, did what is now one of the most important experiments in all of biology, because they showed that this transforming agent in Griffith's experiments was DNA. Okay? How did they do that? Important point. So during the lectures, there will occasionally be a box like this in red that says DNA-I, something, something, something. So that's just a pointer to you that when you get home, you can take a look at your DVD. And in this case, there's an interview with McCarty, okay, the last author on this experiment. McCarty died about 5 years ago. He was well into his 90s. So if you think about this man's life, okay, so he was born at about the beginning of the 1900s when genetics was basically starting, when Mendel's experiments were rediscovered. When he was a young man, he did one of the most important experiments in biology showing that DNA is the agent that is what is inherited. And then he lived for another approximately 60 years to see the amazing advances in biology that have led us to today. So in one man's lifetime, you go from, basically the beginning of genetics to where we are now. It's quite amazing in terms of how much knowledge has accumulated during that time. So what did these guys do? Oh, the other thing I want to point out is, Columbia, Rockefeller, whatever. McLeod was head of microbiology at NYU. Okay, so yay, NYU. Okay. Here's what they did. Started out exactly the same as Griffith. Smooth cells, stick them in a test tube. Okay? Now, instead of just heating them up, what they did is they did a bunch of sort of biochemical trickery and extracted the DNA out of this mixture. Like I said, it's not that hard to get DNA out of a mixture. All it takes is a margarita, right? So they have DNA in a test tube now. The only thing is it's not hard to get DNA out of a mixture, but it's hard to get pure DNA out of a mixture. Okay? Usually when you get DNA out of a mixture, it also contains some amount of contamination by protein and other things. Okay? So, but for now, here's the experiment. Take the smooth cells, get the DNA out, understanding that the DNA might be contaminated with other things. Okay? Now, they can then take the DNA, mix it with rough cells, and then plate it out on a plate. Now, you'll notice that they skipped the mouse part, okay? Which is probably a nice thing to do in terms of humane treatment of animals, right? And it turns out you don't need the mouse, right? So if you take rough cells and mix in the DNA from smooth cells and just plate them back out on one of these plates, then some of the cells will still be rough, but some of them will be smooth. Okay, those are the transformed cells. The reason you only get smooth cells out of the mouse is because those are the ones successfully infecting the mouse, and so when you go back sometime later, those are the only ones that are still there. Okay. Now, they have this problem, right? They haven't proven, let me go back. Oops, sorry. They haven't proven that the transforming principle is DNA yet, because like I said, the DNA might be contaminated with protein, so it still leaves open the possibility that it's DNA or protein. Okay? So how do they deal with that? What they do is they do the same thing, get the DNA, and then treat it with an enzyme called protease. So in biology, if you ever see an enzyme, a name of something that ends in ase, it means it's something that degrades whatever the front part is. Okay, so a protease is something that destroys protein. A DNase destroys DNA. A lipase destroys lipids. Got it. Great. All right. So they take this mixture, they treat it with a protease to get rid of the protein so that all that's left is DNA, mix it with the rough cells, plate it out, and you still get the smooth cells. Okay? But the real kicker is when they do the experiment, they extract the DNA and they add DNase. Okay? When they add DNase first to this mixture, destroying the DNA in this sample, mix it with the rough cells, they don't get any smooth cells anymore. Okay, so that's the experiment that shows that the substance in this mixture that's causing the cells to transform from rough to smooth is DNA. Because when you take away the DNA, you don't get the transformation anymore. Okay? So that's, again, what I would call an elegant experiment. It really just sort of cuts to the heart of the matter using the tools available at hand and had a nice interpretable answer. The only problem was that nobody believed it. Okay? So here's what McCarty had to say about their experiments. So this is a quote from him. He wrote an article in 2003 about this stuff. He said, our findings continue to receive little acceptance. I mean, I just went through it. I just showed you how, you know, the answer is obvious. They proved it, right? But here's what he says. Our findings continue to receive little acceptance for a variety of reasons. The most significant being that the work on the composition of DNA, dating back to its first identification 75 years earlier, okay, back to Miescher basically, had concluded that DNA was too limited in diversity to carry genetic information. Everyone had this preconceived notion that DNA wasn't complicated enough to be the genetic material. Even those biologists who had considered the possibility had dropped the idea, and the prevailing dogma was that if genes are composed of a known substance, it must be protein. Okay? And so the reason behind that is that, as I said, DNA has 4 letters, A, C, T, G. Okay? It has this very sort of simple structure, apparently. Protein has 20 letters, as you'll see in a later lecture. It was already known to do complicated things within the cell. Enzymes like protease are made out of protein. The structural components of cells were known to be made out of protein. Everyone had a lot of reason for expecting protein to be doing complicated things within the cell, and they had no reason to expect that DNA was. So they thought maybe DNA was a scaffold on which the important proteins were sitting, but DNA wasn't really doing anything important in chromosomes or genes. Okay, so that's what they were up against, and that prejudice about what the answer to this question would be was so strong that even though they did this great experiment to show it was DNA, people still didn't believe them. Okay, here's what Avery had to say. You know, so Avery was another guy in the same experiment. This is a letter he wrote to his brother at about the time they were doing these experiments. He says, he was a little bit more emotional about these things. He said, for these past 2 years, I've been trying to find out what is the chemical nature of the substance in the bacterial extract which induces this specific change, which induces the transformation. Some job, full of headaches, heartbreaks, but at last, perhaps we have it. So, you know, this is a real sort of guy who's really feeling it. But today, it takes a lot of well-documented evidence to convince anyone that the sodium salt of deoxyribose nucleic acid, DNA, protein-free, without any protein, could possibly be endowed with such biologically active and specific properties, and that is the evidence we are now trying to get. It is lots of fun to blow bubbles, but it is wiser to prick them yourself before someone else tries to. What does he mean by that last sentence? What does he mean by blowing bubbles?
I mean, it's fun to do like experiments, but you should test them yourself before other people try and find solves.
Yeah, and to the extreme, right? So not just doing the experiments, but doing the experiment after the experiment, and another experiment, trying to show beyond any shadow of a doubt that you're right. Okay, and even though they did that, even though they were so careful, people still did not believe that DNA was the genetic material. And if you read textbooks now, it's not this experiment that people point to as the definitive proof, even though now I think it's fair to consider it definitive proof. People point to an experiment almost 10 years later by Hershey and Chase as the definitive proof that DNA is the genetic material. Okay, so it took another experiment by another group of people to show that DNA is the genetic material. And one of your study questions for today goes into what that other experiment was. Okay, and so you'll read about it a little bit. Okay, back to DNA. So we know it's the genetic material now. If we believe Avery, McLeod, and McCarty, or we waited 8 years to believe Hershey and Chase, we know DNA is the genetic material. We know what it's made out of. It's made out of adenine, guanine, thymine, and cytosine. And we still have to figure out, well, how it actually stores information. Okay? So again, take a look at your DVD and that section on the 4 units of DNA. And so now what we're going to talk about is how you go from the chemical structure, the physical structure of DNA, to information. Okay? And an important clue came from a guy named Char. Okay? And it's now encapsulated in what's usually called Char-Gauss rule. So this is he. This is Erwin Char. Despite the appearance in this photo, he was not working at a diner in the 1950s. He was actually a scientist. Okay? And he discovered something very important about DNA, that if you took the DNA out of a species, say a human being or a bacterium or a plant or whatever, there was this rule that always applied. The amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. Okay? Now, if you read this part in your textbook, it'll also add this equality as part of the rule, that the amount of adenine plus the amount of guanine is equal to the amount of cytosine plus the amount of thymine. Okay? But this is not necessary. Why is this not necessary? Right, because if this is true and this is true, then this has to be true. Okay, so you don't need to say that. As scientists, we like to just sort of give the minimal explanation of something. So this is the rule. A equals T, G equals C. Different organisms had different amounts of each. Okay, so here are some examples. This is a plant that's commonly used in a lot of biological research nowadays. It's called Arabidopsis. And if you go and measure how much adenine it has 32% adenine. How much thymine does it have? 32%. How much guanine does it have? 100 minus 32 divided by, what's the number? 18%, right? How much cytosine does it have? 18%. Okay, so if I tell you the amount of adenine, you can figure out everything else just from these two equalities, because it has to add up to 100%. Okay? Humans, we have 29.5% adenine. Therefore, we have 29.5% thymine. Therefore, you can figure out how much guanine and cytosine we have. Saccharomyces cerevisiae, common baker's and brewer's yeast. Okay, so this is the species that's responsible for beer, bread, wine. Okay, 31% adenine, 31% thymine. How much guanine? 19%. How much cytosine? 19%. Okay, so this is Char-Gauss rule, that it always works out that way. A equals T, G equals C. And you can hear from the man himself on your DVD, and they have a little segment on the ratios also. Okay, so that was an important piece of information because it said there was some regularity to DNA. It wasn't just random A's, G's, C's, and T's, but there's some matching. A's always equal T's, G's always equal C's. Next clue came from a different type of experiment in the early 1950s called Xray crystallography. Okay, and it was done by a scientist named Rosalind Franklin, who has sort of a sad history in biology because she was treated like a lot of women were and are unfortunately treated, and that is her intelligence was put down, her results were stolen, and the credit she deserved was taken by other people. Okay? And so what happened in particular was that she was doing experiments. She had crystallized DNA, okay, similar to what was in those necklaces, right? She had made crystals of DNA. And she was using this technique called Xray crystallography, which I'll explain in a second, to try and figure out what the three-dimensional structure of this molecule is. Okay? we can't look at it under a microscope. It's way too small. Okay? So in order to study it, you need some indirect way of trying to figure out how it all pieces together. Okay? So she was doing these experiments, and her colleague took her pictures, which came out of her experiments, and showed them to two guys named Watson and Crick. Okay? And so Watson and Crick were basically using her data without her permission. And that led them to a very, very, very important discovery. And then Watson, in particular, wrote books afterwards about the whole experience. And really, it's one of the classic examples of sort of misogynist literature. Okay, it's his autobiography. He talks about Rosie Franklin. He calls her Rosie. No one ever called her Rosie. Okay, you can see. So he sort of found ways to keep putting her down, and diminishing her contribution. And the other unfortunate thing is that she got very ill and died very young. And so when Watson and Crick won the Nobel Prize for their discovery, which we'll get to in a second, she was left out because you can't win a Nobel Prize after you die. There's a clip on the DVD that says "Interview with Rosalind Franklin," which obviously could not have happened because she had passed away a long time ago. But it's letters I think she had written to her sister that are read. And I think she's a very sort of interesting figure in the history of biology and science in general. So what did she discover? Well, she came up with a piece of evidence that really was essential for Watson and Crick to figure out what the structure of DNA was. Okay? And what she generated was this picture. Okay? And it's hard to explain how this picture is sort of the essential clue to the secret of DNA. Okay? But it was. On your DVD, there are a few clips that I want you to look at because they actually show you what an X-ray crystallography experiment looks like. There's actually sort of videotape of it, and there's explanations of it. But basically the idea is if you shoot an X-ray beam at a crystal, so a crystal is a repeating structure of some substance. If you shoot an X-ray beam and watch where the beam sort of splits after it hits that structure, you can infer things about what it hit, okay, and what shape the thing was that it hit. And to a crystallographer, if you see a pattern like this, within a microsecond you say helix. Okay? Now I'm not going to explain to you why when you see something like this you say helix, because it's actually very, very technical. But that's sort of what I want you to understand, that this picture really just shouts helix to a crystallographer or a biochemist. And so that's what Rosalind Franklin discovered, that the DNA was somehow a helix. Okay? And so I want to play for you how all of this information came together. Char-Gauss ratios, what we knew about adenine, thymine, guanine, cytosine, and the fact that you had a helix. Okay, so this is Watson describing sort of the process of discovery that went into figuring out that DNA is a double helix.
The Cavendish shop was to build us some tin models, and that took too long. And finally in desperation, I made some out of cardboard. I began moving them around, and I wanted an arrangement, where I had a big and a small molecule. And so, how did you do it? Somehow, you had to form link bonds. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh. So then I went to the pair, and I wanted this nitrogen to point to this one. And I went like this. Whoa, they look the same. And you can put one right on top of the other. We knew he could just, even if we go up to the ceiling, we're building a tiny fraction of a molecule. Hundreds of million of these base pairs in one molecule, all fitting into this wonderful symmetry which we saw, the morning of February 28th, 1953.
So, if you ignore the hokiness for a second. So, he was saying you had to get one of these to point to one of these, and you had to get one of these to point to one of these. What was he talking about there?
The bond.
The base pairs that correspond to each other.
So which ones in particular?
A and T.
Right, so you had to get an A pointing to a T, and you had to get a G pointing to a C, because that's what makes Char-Gauss ratios work. That's what makes the rule work. If you always have A next to T, and you always have G next to C, then you will always have the same amount of G as you do of C, and you'll always have the same amount of A as you do of T. Okay? So that's what made Char-Gauss rule work. And then putting the bases in the middle and that backbone of sugars and phosphates on the outside gives you this helical turn, okay? And two helices winding around each other. So you're going to look at a few other clips related to this. The second one's interesting, but it doesn't really explain why he's talking about the secret of life. So the story goes that they figured this out and they realized they were right. You put this paper model together and it works, and so they knew they had basically figured out what everyone was hoping to figure out, which was, what is the structure of DNA. And they were so excited about it that they did what everyone who's excited in Britain does, which is they go into the closest pub, right? And apparently Crick walks into the pub and just shouts out, "We discovered the secret of life!" And everyone just, celebrates with him, not even knowing what he was talking about. So Watson talks about that a little in that second clip. The other clip that you're going to look at is really interesting too. Where do I have it? Here, this one called triple helix. So just as every scientist expected protein to be the genetic material, if you had taken a bet before the structure of DNA was solved, you said, what scientist is going to figure out the structure of DNA? 90-something% of the people would have said Linus Pauling. Okay? So Linus Pauling is a famous chemist, biologist of the last century. He's especially famous for winning 2 Nobel Prizes. Okay? He won one for his scientific work and one Nobel Peace Prize because he was a strong proponent of doing away with above-ground nuclear testing. So everyone knew how smart Pauling was. Everyone knew he was working on it. Okay? And so everyone assumed that if someone was going to figure out what the structure of DNA was, it would be Pauling. And so this clip basically shows Pauling's best guess of what the structure of DNA was before Watson and Crick told everyone. And he was so wrong. And it's kind of illuminating because it sort of humbles you, right? It's a way of reminding you that things that seem obvious at one point were not at all obvious, and even the smartest people in the world at the time couldn't figure them out. Okay. All right. So the next thing I want to talk about is the last part of that clip of Watson showed this thing sort of growing up until this really, really, really tall ceiling, and he said, go way beyond the ceiling because you need 100 million of these things stacked on top of each other to make one human chromosome. Okay. In total, we have 2 pairs of each. We have a pair of each chromosome, so we have 6 billion letters of DNA inside each one of our cells. 6 billion letters. That's what's shown here. Okay, so MBP is millions of base pairs. Okay, so 6,000 million base pairs is what humans have of DNA. That's a lot of DNA, right? One of your study questions is figuring out how tall that string of DNA would be, actual sized, if you took the DNA just from just one of your cells and stretched it out. Okay, and you'll be surprised at what the answer is. Because if you do that, and then you think about, well, if you stretch out one cell and your body has maybe 10 or 100 trillion cells, how much DNA is in you and how far that would actually stretch. So he was not sort of exaggerating when he said, you know, it would just keep going and going and going. And if anything, it was an understatement how far that ladder of DNA would go up into the sky from just one of your chromosomes. Okay? And one of the things we know now is how much DNA is in a lot of different organisms. And so here's just a quick rundown of some of the more famous organisms. So E. coli, that's this, right? Each cell, the diameter of each one of these cells is about 1/1,000,000th of a meter, okay? Each cell contains 5 million base pairs of DNA, okay? Baker's yeast, brewer's yeast, each cell, 25 million base pairs of DNA. The fruit fly, again, common laboratory organism, 330 million base pairs. Humans, 6,000 million, 6 billion base pairs of DNA per cell. Okay, and if we just went up to here, this would probably sort of make sense to you, right? We're sort of increasing in complexity from a bacterium to a yeast, which is also a very simple organism, but is more like us than it is like a bacterium, actually. Up to a fly, which seems in some way more complicated than a yeast, but not quite as complicated as us. So here are some surprises. Wheat, okay, a plant. 32,000 million base pairs. Okay, 32 billion base pairs of DNA per wheat cell. Okay, I guess that means that wheat is, you know, 5 times more complicated than we are. Here's another one that's sort of humbling, right? Flatworm, Otomestoma, right? 40,000 million base pairs. Okay, 40 billion base pairs of DNA per cell for this lowly flatworm. Okay, so something's going on here. Okay, first of all, it looks like maybe there's a general increase in the amount of DNA the more complicated an organism gets, but there's definitely not an absolute correlation, or our notion of what makes something complicated is way off. Right? We like to think ourselves as the most complicated. And in which case, you know, these numbers are just really surprising to us. The record holder, which I looked up for an animal, so is actually a lungfish. Again, not the kind of animal you'd guess is the most complex. 130,000 million base pairs. Okay? 130 billion base pairs of DNA per cell. Okay, so this means 2 things. One is that, you know, maybe not all of the DNA is important because, you know, maybe we have as much DNA as we have, and adding this much more DNA doesn't really make you more complicated as an organism. Maybe that's true. We'll see in an upcoming lecture how much of this is actually meaningful. Okay? It turns out that in some ways we're not that much more complicated than flies. We just have a lot of extra junk in our genome. The other thing that this raises is how you get all this DNA into each cell. Okay? Because think about it. Let's say you had a really long ball of string and you had to carry it around with you. You're not going to carry it around as this string that you're just sort of trailing behind you, because very quickly it would get tangled up. It would, you know, trail behind you a long distance. So the right thing to do is to sort of wind it up and carry it as a ball of string. And that's what your cells do to their DNA. Okay, so DNA inside your cells is packaged. And this is a diagram of sort of how that packaging takes place. It happens in sort of successively more and more compacted states. Okay, so you start with just this double helix of DNA, right? And that's wound around certain proteins called histones to make nucleosomes. Okay, so this unit of DNA wrapped around histones is called a nucleosome. And this is sort of one level of compaction, that you have what's called beads on a string. That you have, instead of one long string, you have a string and then a winding and then a string and then a winding and a string and a winding. Then those wound parts actually stack up onto each other. And then those stacked up things fold in and in on each other. And then ultimately, you have this sort of compacted thing that looks like a chromosome. Okay, so I want to show you from your DVD an animation of this, which I hope will help to make it more clear. So, and I'm turning the sound off because it actually gives you the answer to one of your study questions. Okay, so here we have just sort of a representation of this double helix of DNA. This is one of those histone molecules, and the DNA gets wrapped around a set of those molecules to form a nucleosome. Okay, so these are what form those beads on a string. See, nucleosome. And then those get packed tightly up against each other. By the way, so these protein molecules that the DNA is wrapped around, this is why it's hard to get pure DNA, right? Because every piece of DNA is actually wrapped around proteins. The other thing is that DNA is negatively charged. These proteins are positively charged. Opposite charges attract, and this is what makes this sort of compaction possible. Right? So you get this structure of things getting packed against one against the other. It's sort of, I don't know if people still do this in summer camp, right? You take a long string and you fold it in half and two people wind in opposite directions and then you put the ends together and you snap it and you get this sort of compacted thing, right? I'm probably aging myself by saying that's what we did in camp. Okay, that's sort of what you get. That's what's called this fiber. And then that compacts even further and further. Now it looks like sort of a telephone cord sort of wrapped on itself. So you get this like sort of coiled structure. And that's ultimately what gives shape to chromosomes inside of a cell. So this is an actual picture of a cell dividing. These blobs are the chromosomes. And what you can see is as the cell divides, those chromosomes become more clear. And then they separate from one another. Okay? These chromosomes go that way, those chromosomes go that way. And then two cells form. Okay, so this is sort of the kind of mental gymnastics you have to go through when thinking about things that are this small, right? The DNA molecule we just can't see. It's tiny. But it's packaged, it's grouped together, and then ultimately you get something that you actually can see under a microscope at least. Okay, so if you want to look at that again on your own, that's the thing to look at. And what I want to finish with today, before I give you your homework, and what we'll pick up again next time, is, so DNA we now know is the genetic material. We know it has this double helical structure that A's pair with T's, G's pair with C's. And immediately upon knowing that structure, that suggested something about how the DNA molecule is copied. Okay, because remember, if it's going to be a genetic material, it has to get copied, right? And so this is what Watson and Crick had to say about it in their paper where they reported the structure. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what do you think they were thinking? That's right. So if basically what you have is a molecule, this double helix, that has redundancy built into it, right? If I give you one half of it, you know exactly what the other half is. If this is AAT, you know this has to be TTA, okay? So in a sense, it doesn't need both sides. It just needs one side, and one side, each side alone carries all the information. Okay? So what they saw was that if you have this double structure and you separate it, then you could reconstitute the whole double structure again just by copying off the strand that you have. Everywhere you see an A, you put a T. Everywhere you see a G, you put a C, and so on. And then you get two identical copies of the thing you started with. Okay? So one of the things we're going to discuss next time is how it was actually shown that that was true. Okay? Because it's one thing to say this immediately suggests a copying mechanism. It's another to prove what that copying mechanism is and what type of experiment you have to do. So next lecture, what we're going to start with is another example of a now classic experiment in molecular biology that, in a sense, now seems like a really obvious result, but at the time was another essential piece of the puzzle that had to be shown. And again, it's one of the most elegant experiments that's ever been done. So that's what we're going to talk about next time.